Center for Advanced Engineering building. If you're from Erie, it was actually the old Boys and Girls Club. So it was originally there before Gannon got the building. But that is number 41 here. Students do have access to it, so there's a little area students can go and congregate in, but the area itself, all of the vegetation is not affected. And then our other learning commons on top of our library. The Center for Advanced Engineering is located on 8th Street, while the library one, 6th and Sassafras Street.
Alright, so going back into like the locations we had, this is the engineering building, which was a lot older. As Libby said, it was there before Gannon actually got the building. So around 25 years old. You can see there the student access, so they can go sit at the tables and stuff. But yeah, the 4 plots there that we tested at aren't actually affected by any like pedestrians. And if you go to the next slide. And this is the National Library one, so on the 3rd floor, more of a rooftop. This one doesn't have any student access. And it's also got the 4 plots there. And you can see the dates there for when we actually did our experiment. So around the end of September, 2 different, like a week apart. And yeah, on the next slide. So yeah, this is we have the same testing techniques across both locations. The aerial fly strip, the ground fly strip, and also the Sherman live mammal traps. And then, go back one. And that's there on the National Library as well. This is our control area, which was just outside of the library there. We just did 1 plot for this, so just the same area of 5 by 12 feet. But there was just 1 plot compared to the 4 testing plots we did on each green roof.
And this is middle one, right?
Middle one does the
Middle one's
Yeah. And this is the Mercyhurst green roof area up here on top of the Zern building here, up right here. And down here on this front line near 38th Street is where we did our control plots, which is also around where the future tiny forest is going to go. As you've seen before, these were both installed in 2010. And here is the main green roof area. And here's the north strip we have that's mostly sheltered in by all the walls. Also, as you can see down here, these are like Gannon's tray green roofs, but because ours is a little more newly installed, you can still see some of the trays there down below. Not the best view, but and same as Gannon, same sampling. 4 plots, 1 vertical, 1 horizontal, and 1 vertical, with 4 plots in each area, so 4 along the strip, and 4 in the main area, alongside the Sherman live mammal traps. But our dates are a little different, a few days off. So we ran to the first day of October for our second one, so about a day or so off. And this here, though, is our main control area, and unlike Gannon, we did 4 plots of verticals and horizontals, and on a much bigger lawn than the little strip they had, but we did not put out any of the Sherman live mammal traps in our control area. Oops, sorry.
So this slide is just showing the insect diversity on the Gannon green roofs and also Mercyhurst green roofs. So a total of the 4 green roofs, just so you can get a better idea of the diversity that we had. Okay.
So this is just comparing like the different orders that we found. They are very similar. Both had a really big like distribution of the Diptera family there, but as you can see, the red wedge compared to the orange wedge, we had on the Gannon roof, there was more of the Lepidoptera, where that wasn't seen on the Mercyhurst roofs. Instead, they had some of the Orthoptera. So that was probably the main difference, but on that, the distribution's pretty similar across both areas. Yeah, this is looking at the lawn control, so,
Locations that the insect diversity differed a bit. And then, of course, Gannon is located down by the lake, so that would have some significance as well, as Mercyhurst having more green space and more foliage around their campus. And then we focused mainly here on the green roof, our main green roof. So again, you can see all around, there really isn't much green space. It's more, like I said, of an urban setting, while if you look, the Mercyhurst one,
Well, the Mercyhurst one here, up here, again, you can see our control site up here, so a lot of green area around it, beyond just the control site. We've got all this green area around it, more green here, a whole lot of little lawns here and here. And up here, you can see the other lawn. Up here, you can see our green roof area up here. And you can see a lot more like suburban green, just generally green area here compared to the Gannon University campus. So just generally a lot more green area. There we go. And as mentioned before, there's future plans to enhance our green roofs, do further studies with seeing if we can get any of the small mammals, or any data on small mammals there. And of course, we couldn't have done this without both professors from Mercyhurst and Gannon. And Nathan and Skylar here, they were some of the original people back in 2020 from Gannon, who did their part in the green roof study. And Zach and Lisa here from Mercyhurst are the ones who actually designed and set up the Mercyhurst green roof back in 2010-ish.
so that is
Questions?
all.
We have a few minutes for questions. Anything?
So you mentioned the impact of winds on your insect diversity and just general count. So does that mean that when you have more wind there would be more or less insect diversity?
So on the Mercyhurst green roof, there's like two different plots. So one is a larger plot that's like really exposed, like the walls near are pretty low, and that's the main roof. And then the north roof is like set a little bit lower. It has like 3 walls surrounding it and kind of a bit of an overhang. So we figured that it'd be a lot easier. The insects would be carried over in the wind and could get stuck on those traps, or we're most likely to visit the more exposed areas, and it'd be harder for them to get behind into like that smaller area with like the enclosed walls.
Okay, thank you.
Anyone else with questions? Yes, Amber.
Have you considered using GIS to map the actual land use around your campuses? Things like green space versus impervious surfaces? That might help explain some of your data too.
Yeah, if we wanted to further this study, that would be a great tool to kind of add on to it and get more solid data for that.
John.
What made you settle on the sides of the area bordered in red? How did you figure that you needed a 0.25 kilometer area?
I'll confess guilt in that. Yeah. I like the level of resolution that could be seen at that elevation where the photo was taken, and that 0.25 kilometers seemed like a nice, even metric area. Draw a box around it. There was no sensible biological data. I'll take the heat on that one.
John.
Did you see any sign of leaf liners or some, non-flying insects or spiders for that matter?
I'm sorry, what was that?
Did you see any sign of leaf liners or caterpillars or spiders or, non-flowering, non-flying taxa?
So we ended up catching one spider. I think that was it though.
Yeah.
Yeah.
Just the one spider.
And this was done in sort of September, mid to late September / very early of October, so it's a little late for caterpillars to be out.
And Mike mentioned that he was hearing crickets a lot on the Mercier's roof, so.
Yeah, you know, they're one late, they were recording.
I just want to say it was a very interesting presentation, and I think it would be, for future plans, be interesting to do it in a different season and see what other insects you find. I think it's very fascinating.
And I'm gonna hop in and say, we talked some more with Sarah Bennett, and Mike, I mentioned this, that Asbury Woods has a green roof, but they were reluctant to let us use their green roof as part of a study. But Sarah says she'll maybe work around that and see, because their green roof is, our roofs are sedums, their green roof is meadow and native plants. So it'd be kind of cool to get a third one in there with a whole different diversity of plants, flora and fauna, and see what they get compared to ours. So we may expand it that way. That's your job.
You know, the Chestnut Street Water Plant has the first green roof in Erie
That's
down
right.
there too, and theirs is like a lawn. I think.
Yeah.
And I think they mow there to keep the. So it'd be interesting to include that because that's so close to the water.
Good idea.
I was going to ask if you took, and you might have mentioned this, did you take any like plant surveys? Because, you know, you're comparing the two locations. If the vegetation is different, then you might be attracting different species of insects with that. Do you have plans to do that, or was that kind of, I know that Steve mentioned it was sedums that were mostly on it.
I didn't hear. Question, Jim?
Vegetation surveys on there, were they very similar, or did you kind of document? I saw you had an aster growing out of one of the panels on the roof.
Yeah.
Yeah, so both of the green roofs are sedums.
Okay.
And we're in the process of counting all the blown-in plants that we have on ours. So there's still quite a bit of data to be collected.
Oh, that's great.
Yeah, and we talked about, like, just before, whether or not we were gonna, like, keep the flown-in plants just for a little more of experimentation, or getting rid of them to kind of level out our green roof and keep that going.
Okay, yeah, interesting. All right, well, thank you very much. Oh, I'm sorry, we still have more.
Are the roofs lit up at all, or is it pretty dark up there? I'm not.
At least in terms of the Mercyhurst roofs, there isn't really any outdoor lighting there.
Oh, okay.
I don't know about the Gannon ones.
Yeah, the only lighting you get, maybe on the Nash Library, because it's open till like midnight, there might be lighting just from inside, because it is just surrounded by windows on one side there from the 3rd floor. So there might be some lighting there, but other than like external lights on that area, there's nothing.
Okay, thank you.
All right, okay, well then, thank you very much, and And as we're transitioning, I'll introduce our next speaker. We have When a Duck Nests on Your Green Roof. And that's being presented by Cassandra McLaughlin from Gannon.
I don't know where it is.
Yeah.
I love it.
Oh.
Let you know who's sitting at that table.
Maybe a little bit of a larger one because the video clips there in it. So let me take it just a moment. Okay.
Visuals are what this also works
yeah.
also,
Yeah.
especially in this case.
Absolutely.
That was something we definitely didn't expect to happen. I'll talk a little bit about that when I get it up.
Bit of surprise.
That was Dr. Robesky actually that found her, so.
Was he like running around the building like?
It took a while.
I know,
'cause
it's
the
got
video
big,
clips in
yeah.
it. And there's the remote.
Okay, perfect.
All right, like you heard, my name is Cassandra McLaughlin. I'm a pre-vet biology major at Gannon University, and I did this research with Dr. Stephen Ropsky, who is a biology professor also at Gannon University. So this whole story starts off quite interesting. I was working on trying to find myself a good research opportunity in the Erie area, and I was really looking at the Erie Zoo originally. But due to COVID restrictions, I wasn't able to do any research there at that time. So I got in contact with Dr. Ropsky, and I said, you know, I'm really having a predicament here, and I need some help. Do you have any ideas? I get an email right back, maybe about 5 minutes later, and he goes, well, you'll never believe this, but I have a great opportunity. I was like, okay, cool. So I gave him a call, and he goes, well, I was weeding on top of Nash Library's green roof, which originally I didn't even know was there. I hadn't been up there yet. So this was news to me, and he would have been weeding right over here on top of Nash Library. And he goes, only about 2 feet behind me was this mallard nested right within the sedum. You couldn't see her. He'd been up there quite a few times, you know, towards the beginning of summer. And this is when we found, just a side note, her name is Willy, so I'll be referencing to her as Willy. And like I said, she was only about 2 feet behind Dr. Ropsky, so he was pretty much almost sitting on her while he's weeding this green roof. And as you can see, our green roof doesn't have any water. You've heard a bit about it. We don't have any water on top of this green roof. We're 3 stories up in the air, and the green roof itself is only 4 years old. So just the fact that Willy took notice of this green space as a safe area to nest was really interesting to us, especially the fact that she can see everybody and everybody can see her. It's a little bit of an interesting area from a behavioral standpoint, and also looking at where ducks would normally be nesting, which would be near water and low to the ground. Willy likes the opposite. Alrighty, and then also another thing to note about our green roof is that it is pans, just because our roof is non-weight bearing, like most of our green roofs in the area, as you've heard about prior to this. So this would be on May 3rd, yes. So May 3rd was the first day I got set up. The day that we actually noticed that Willy was there, I'm sorry, not May, June 3rd, May 23rd is when we noticed that Willy was there. And then May 24th, we had actually heard that there were no eggs in the nest, or that there was a potential for there not to be eggs in the nest. And we just had actually heard about that from our library staff, who play a very large role in this project. But I rushed over there on June 3rd, and this was our first day set up. So we started right away with getting cameras going. And this is actually a GoPro stand. You can see over here that that's right where Willy's at. So we're just trying to see, you know, what movement can we see? What behaviors is she exhibiting? Is she showing us signs of sitting on eggs? And so on. So this is really, like I said up here, a race against the clock. We don't know if she has eggs. We don't know if she's leaving the nest. And you know, we're trying to figure out, why is she here? So we get our 3 camera varieties that we worked with for a majority of this time period. So we had the GoPro originally, which unfortunately its battery didn't last as long as we would have liked. So we moved on to our next options, which would have been the CASA surveillance camera. So it's like a home surveillance camera where you'd be able to see everything 24/7. Unfortunately, that only worked for a little while, so our best bet right here was our Reconnects trail cameras, which we actually sat out on the green roof in a very stealthy way, and we'll see those pictures coming up here as well. And aside from just our cameras and looking at all of the information that we were gaining from that way, we were also working, Dr. Ropsky and I, for an hour from 2 to 3 o'clock every day. And then we also had, like I said, they'd come into play, the wonderful library staff that was there during the summer. So from 8:30 a.m. until 5:30 p.m., beginning on June 9th, is when our library staff would go on top of the Nash Library and take a look at Willy and see if she was leaving the nest, which noted on all of their time charts is that she never left the nest. And we were really trying to figure out, is she still leaving to get energy to take care of her young? Turns out that she wasn't, based off of their observations and also our cameras, and also what Dr. Ropsky and I were collecting daily while we were there watching her. So right here you see, this was our first documented egg sighting, officially, where either Dr. Ropsky or myself had finally said, yes, there are confirmed eggs in there. And we could only see about 2 or 3 eggs. And from that point on, we knew that mother ducks typically sit on a nest for 25 to 29 days. So we didn't know how much time we had from this point onward until hopefully the babies would hatch. And then here, while we were working on trying to figure out when these young would finally hatch, we were also looking at the behaviors of Willy. And this was one that was really interesting for us. Not only was she exhibiting a panting behavior, which is when her beak would open and close very slowly. She was trying, it was very hot up there, so she was still trying to make sure that her temperature was okay. But right here is actually what we were calling at the time chattering. But with later research, we found out that that was actually vocalization. So the mother ducks will do this in order so that their young can hear their voice, learn it, and then also toward later stages of development begin to vocalize back to the mother. And this will also later on help them synchronize when they will all hatch. So that's a very interesting part of this whole thing as well. So back to the trail cams. This was much closer to the most important part of this whole thing. But this is our Reconnects trail cams, and we put them on the roof beginning on June 11th, and they remained on the roof for 8 days, and they were on day and night, so they have night vision. We would be able to see if she was leaving the nest or not. And as you can see, we have some friends who would visit us. There was about almost 300 pictures of our pigeons and other random movements, but no Willy, and you would have seen her. She would have flown up from about right there, or if she was even looking around the green roof in general for food or water. So on Father's Day, we had the unexpected hatching. This was, you know, this was much shorter time frame than we were expecting to work with. Luckily, Dr. Ropsky took time out of his day with his family on Father's Day to go and check on Miss Willy, and wouldn't you know it, a baby pops out from under Willy. We have ducklings. And of course, I get a call, and I can't remember where I was at, but anytime he called me, I knew something was going on. He goes, Casey, tomorrow we've got to make sure we get these babies off the roof. And that was actually another issue we encountered was, do we let nature play its course, or do we take action and get the babies off the roof? Because you have to remember, they're 3 stories off the ground, and they don't have water, which is essential to their first couple of days on this earth, and we want to make sure that that worked out all right. So on June 21st is when, the day right after the babies hatched, is when we got together with Wildlife in Need in order to get Willy off the roof. And we also had a great community effort from the Gannon students as well. Some people just that we needed to be there because we didn't know how many babies we were dealing with or how Willy was going to react to us trying to relocate the babies. So here she is looking a little more rough than she normally does. You can tell that her feathers are ruffled out. She's really just trying to keep the babies close to her. She was used to us being there, which made it a little bit easier as far as getting the babies. But it was still an uphill battle. So we ended up seeing that there were 5 babies that had actually hatched. And the reason she's walking around now is because our Wildlife in Need experts were coming onto the roof to try and fence in the area around her. Unfortunately, Willy is very smart, so she began to try and find a way off of the roof or a way away from the people who were trying to get close to her young. And right here, yep, is when we finally caught Willy and the babies. And you see our Wildlife in Need expert right there. And then also, that is Gina in the tie-dye. She is where the babies and Willy eventually ended up for their rehabilitation period. So another important thing that happened was that we actually had to put Willy and the babies in 2 separate cages so that there weren't any injuries of Willy herself and then also the young, because they are very fragile at this point in time. All right, and you see you're getting her in there. And then right after this, you'll see them collecting the eggs from Willy's nest. And before they began to collect the eggs, our experts here were actually listening down into the ground to see if they could hear any vocalizations coming from the eggs so that they knew what they were going to be dealing with as soon as they got Willy rehomed. And they're going ahead and putting them in the box with the sheet after they have decided that they haven't heard anything coming from them, but they're still going to bring them with, as well as the pieces of the nest that they're actually able to pick up and put in the box. This will allow Willy a little bit more time to try and incubate these eggs and get them to hatch. But at this point, it is a little bit too late. And then next, finally, this was the most relieving part, is when we finally got them off the roof. And one thing I did forget to mention is that while they were actually on the roof trying to catch these guys, Dr. Ropsky and I had come down all these stairs. This isn't the first time we'd been down these stairs today. We'd rushed down these stairs with 2 bed sheets, just in case the babies were to fall off the side of the building. So we had little parachutes and all of that. And you see us, we're just getting to the Wildlife in Need vehicle at this point in time. But once Willy and the babies were taken by Gina, they had actually gone to Northeast to live with her. She works with multiple different birds. That's her specialty. She works with wild birds and waterfowl and things like that. So they would actually remain with Gina until late August. Unfortunately, during this time period, Willy did manage to escape. When the babies were a little bit older, when the ducklings were a little bit older, she had actually figured out that when the volunteers would turn away to get water for her, they would leave the cage unlatched. It was closed, but unlatched. And she would actually back herself up to the back of the cage, and she rammed it and flew out. The babies tried to follow her. Unfortunately, they didn't know how to fly yet. But all 5 babies ended up being all right. Unfortunately, like I said, there were only 5 babies, which meant those other eggs did not hatch. And what she did at that point in time when she realized they weren't vocalizing back to her is she had actually smashed the eggs and ate them to take back that energy that she had allocated into their development at the beginning of her incubating them. So here's the final part. This is kind of the sweet ending to the story, is that all 5 babies were released on August 31st in Union City. And you will notice that there's 6 of them here. That is because there's another orphaned duck here, not of the same little brood that was there before. And then I did skip a picture, but they're actually in an area with multiple other ducks to be able to teach them teach them their reins. Here's how we get a grip on the wild. And then I just want to give a big thank you to Janet Laboda, who is the library staff that I alluded to. She was the main lady in charge of all of those time charts, and then with the other Gannon employees there to help out with us. Sue with WIN, which is the Wildlife in Need for the capture and rehabilitation. Gina, as I mentioned, was their caretaker until the release. Austin Froelich for the photos and video content, probably about 90 or more % of it. And then also Sarah Till, who helped us get a lot of things together, whether Dr. Ropsky and I couldn't be there on a given day. We made sure that things were taken care of. But are there any questions after all that?
Were they all female, or hadn't those ducks just developed their
They did end up developing as all female, and I'm not sure if that had anything to do with the conditions that they were under, but all the ducklings ended up being female. Yep.
Any other questions? Oh, yes. Did you put a tag on Willy?
We did not, and that's actually something I forgot to mention. I'm going to put this note down. Okay. We are actually very excited to see if she'll come back next year. You never know. To see maybe will she return to nest where she was at, or was she too disturbed after we removed her? We're not sure, but I wish we had tagged her.
So when I call a game commission, I I said, we have a duck on our roof, do you want to come and do stuff? They said, we let nature take its course.
Yep.
So they did not even want to come and mark or tag it, and I can't tag ducks, so I don't know. So we're curious to see if Willy comes back next year. So was this a successful nest? I don't know. In her mind, maybe. In our mind, I don't know. We'll see.
I'd say she got 5 babies. It was good.
So how long was she up there without leaving?
So she was up there from June 3rd, that we know of, which would have been our first day, kind of getting our cameras ready, all the way until June 21st.
Okay.
Yep, from till the day that we moved her. So she hadn't moved at all. And that was our main thing is she gonna move off this nest? Are we gonna see if she's still leaving to get resources for herself? And no, once they go into that brooding stage, they don't move. They stake it out. Yep.
Okay.
So the conditions on the top of the roof, were they much warmer or hotter? She seemed very exposed
Yes.
compared to like a typical nesting site that they would select.
Right, there was a little overhang, but we actually think that she picked this area just because of, it seemed secluded to her, so there was the walls and everything, and like I said before, we were noticing a panting behavior coming from her, because a lot of these days were over 80 and sunny, as I had noted in all of my journals, but that's why she was panting and all that, but.
I just want to point out that when you showed that video with her panting, I could see bugs zipping by.
Yeah.
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 She's like a cottony with her mouth open.
Okay, we have time for one last question. Okay, right.
Follow up to the time frame that she was on the roof. So during that period while she's nesting, does she need to intake food or water, or does she basically just survive?
No, normally if she would have been a little bit earlier on, she may have still been collecting resources in order to sustain herself and then also sustain her energy to be able to incubate the eggs. But typically when they're, later in their incubation process or brooding process, they've already collected enough energy to be able to allocate more toward reproduction. So, yeah.
Thank you very
Yep,
much.
of course, thank you.
Sarah, is yours the?
RSC migration monitoring.
There it is. All right. Hang on, let's get you
Take
into-
off my mask for talking.
Yeah. Okay, so next we have Sarah Sargent. She is with Erie Bird Observatory, and she's going to be speaking about monitoring migration and tracking movements of songbirds at Presque State Park.
Great, this is a change of pace from the last talk. That was very cool, I have to say. But this is going to be very different from that. So, migration is a tremendous phenomenon, annual migration on the part of songbirds, and also waterfowl, things like mallards also migrate. And so one of the things that Erie Bird Observatory does is that we try to monitor the migration. That is, how many birds, what species, when are they coming, lots of different facets of the actual migration phenomenon. So I'm going to talk about 4 different types of work we have done here. But first I want to make sure you truly understand and appreciate the magnitude of the phenomenon. I'm going to talk, let's see, about, where am I? This is just an illustration of what really happens during annual migration by songbirds. This is a species of bird called the black-polled warbler that weighs about 12 grams. And in the wintertime, you can see all that yellow area in northern South America. That's where they spend the winter. And I don't know if I'm actually going to be able to make it start. This is a video. Is it going to start? Yeah. So this is an animation of the abundance of black-polled warblers through the year. They are going from South America all the way up into the spruce forests of Canada to breed. And that's what they do every single year. Their entire population spends the winter down in South America, but they only reproduce when they're in North America. So they have to go all the way up to these spruce forests. They eat things like spruce budworms and lots of insects up there. And then they turn around and fly all the way back. So this is a 12-gram bird. So this is what is so cool about migration. These are tremendous athletes, these birds. They're tiny, but they're going huge distances, and they know how to do it. And so they can navigate and they get back and forth. So it's just incredible phenomenon that they are able to do this. And there are dozens and dozens of bird species that do this. Not all of them go all the way to South America. A number of them spend the winter in Central America or the Greater and Lesser Antilles, the Caribbean islands. And some of them only go as far as the southern US. But nonetheless, this is a tremendous natural phenomenon that happens every single year. And I think many people are just not fully aware of that it happens. So, oops, there it goes again. Oh, how do I make it go? There we go. So the question is, why do we want to monitor songbird migration? Sure, they're doing it, so what? Well, the thing is that they are declining. And this is a paper that came out, this is a couple factoids from a paper that came out about 2 years ago in Science magazine, and using a lot of monitoring data that's been collected on birds over the last 50 years, estimates of declines in migratory bird populations are that approximately there's 28% fewer migratory birds now than there were 50 years ago. So this is just illustrating with the case of the Baltimore oriole, which is a migratory songbird that spends the winter in Central America. 2 in 5 Baltimore orioles are not here anymore. 50 years ago, there would have been quite a few more than there are now. So basically, monitoring is what gives us this type of data that allows us to do these calculations, to understand that we are losing things. And the other component of it is that migration is, there's been enough work done on population survival and mortality of songbirds through marking that we know that migration is the most, and also it makes total sense, migration is the most dangerous portion of their annual cycle. We have good estimates of survival during the breeding season, good estimates of survival during the non-breeding, the wintering season, the 2 static phases of their annual cycle. And so by different, and then we can get estimates of their overall survival. And so the difference means it is what we estimate as the mortality that happens during migration. And so that's why I find migration totally fascinating. And in terms of conservation work we can do, I really think migration is a critical time for these birds. And we want to try to do what we can conservation-wise, which is why so-called work here at Erie. All right, enough about why we do it. What do we do? Actually, okay, a little bit more on why. Basically, any kind of migration barrier, like large bodies of water, when you get barriers in the way of migration, the birds pile up and have to divert around it, or they have to, they squeeze in to smaller amounts of land, they get compressed. And so this habitat, land that's adjacent to any of these migration barriers, is really critical. Gets heavily used during migration periods. And of course, that's, oops, here we go. I forgot I had this animation. That's what happens when you don't practice your talk quite enough. Okay, there's hybrid densities in the land adjacent to the fallouts. I don't know if you've heard the expression fallout. That's when a whole lot of birds suddenly land, and because of a changing weather typically. The availability and the condition of that habitat that gets so heavily used is really, really important. And it's even worse if in an urban situation like the city of Erie that's right on the edge of the water, the birds have a hard time making do. They have to go around that. It's not good habitat for them. And so this leads to some conservation issues for migration, and how can we cope with that and make it easier on the birds? Okay, here's what I'm going to talk about in terms of what your Bird Observatory does in monitoring. I'm going to talk briefly about 4 different types of work that we have been doing, 2 of which we only started this year, nocturnal flight calls and morning flight counts. Misnetting banding's been going on for a long time, and then this tracking of movements of individual birds with little radio tags is something I'll end with. First of all, I'm going to talk briefly about nocturnal flight calls. Almost all these songbirds migrate at night because it protects them from predators, and also there's been some work on the aerodynamics. They're more efficient flyers at night. And so the nocturnal flight calls, there's been a lot of kind of work over the last 20 plus years really about trying to detect these nocturnal flight calls. The birds make little calls, soft calls to each other because they're in groups as they fly at night, and they try to stay in touch with their flock mates during the nighttime flight. And so you can record these and get an estimate of what species, and you can identify species if you're really good. You can identify by ear by hearing them. But you can get the volume and the species composition of flocks going through if you can record them and then analyze these recordings. And so, Joe Geikis is a guy who lives in State College, and he's really, really interested in doing this. He gave me a new bucket microphone to put up on the roof of the tower. So this went up. There actually had been a bucket microphone. Some of you may have remembered this going up back when the building was first built. There was auditory recordings being done with a team, including somebody from Powder Mill, Avian Research Center, and I guess Ken Anderson from Gannon. And they put this old bucket up there. I found it. It was still up there. So I took it away, and we replaced it with a new bucket microphone in early April. So we started getting recordings. There's a cord that goes down from the bucket microphone and plugs into this laptop that's in the floor right below, and that is recording on the hard drive all the sound files. Once an hour, it closes the file, starts a new one, and it stores it. So we had this going. We ended up having some technical difficulties, and we thought it was recording all the way through the end of May. But when we went up there, we found that there were some problems. Apparently the sound card was bad or something in this computer. And we only ended up having 17 and a half nights worth of recording from early April through May 17th is when it died completely. So we don't have complete records for this spring, but it's up there right now. Hopefully we'll get some good records from the fall. In the sound files from those 17 and a half nights, we did have over 5,000 sound detections. There's a lot of fancy computer software now for identifying sounds. Many of those are not bird calls, but many of them are, and we have yet to really fully analyze all the sounds. So we're still working on that part of it, but hopefully we'll have that out pretty soon. So that's one component, and sure enough, lots and lots of birds flew over and gave calls, and we picked them up with the bucket microphone. There'll be more to come on that. The second thing we're doing now that we hadn't done before is morning flight counts. I said a lot of these songbirds migrate at night. However, some of them continue into the morning a bit. They're not quite ready to sit down. Dawn starts, the light gets up, rises, but they continue to move to some extent immediately following dawn. Then there's a whole set of birds that are actually diurnal migrants. And so that includes a lot of the blackbird species, and also blue jays and robins. And so Jason came and started working with the Erie Bird Observatory doing the water bird counts in the early spring, but he really wanted to do morning flight counts from the tower because it puts you right up into this perfect elevation for a lot of birds that are going right by. And and also he's an expert at morning flight counts, turns out, and he's able to count, identify and count like 12 species simultaneously by scanning around. He uses his binoculars, and he has a camera where he can capture records of birds. So this is a picture he took of an oriole flying by from the platform. So you can see it's right at eye level. And what he was finding was that tremendous number of birds do go by, and he counted them no matter what direction they were going, but he did record them based on which direction they were coming from. The majority of them, as you can imagine, were basically southwest to northeast, and what they're doing is following the bluff line. So there's a bit of an updraft, I think, from the bluff, and the birds are just following the edge of the lake, the line of the bluffs, and they go right by the tower, and you can count them from up there. So that's what he did. He had over 300,000 birds counted. He was up there for 38 days, about 3.5 hours per day. He quickly learned that some days were not nearly as good as others, and I'll show you that in just a second. Some days were super big. The biggest day he had was March 9th, 43,000 birds he counted, estimated in groups of 100 and whatever. He did stay up there longer that day because the birds kept moving. But he had 10 total days with more than 10,000 birds. And here's the sequence. You can see that there are clusters of days that are when there's a lot of passage happening. And those are days with south winds, okay? The birds are smart. They're not going to battle a headwind. They really surge on days with tailwind. And so that's what happens. So the timing of these clusters is related to front movements going through. And the birds are using that, taking advantage of the weather. But it's also kind of interesting. You can see that later on in April and May, and this is when we usually have our bird banding season going, is April and May. A whole bunch of migration has already happened. And Jason says it really probably is even happening at the end of February with some of these birds. So we're going to start earlier next year. That's our intent anyway. But I think what's happening is that the ones that are truly nocturnal and want to sit down in the daytime, get down out of there, that's what's happening over here. These other species have already largely moved through. And these are the more diurnal migrants going through earlier. And just to give you a species composition, out of the 322,000 birds approximately that he counted, the big flocks were red-winged blackbirds, common grackles, mixed blackbird species where they were a little too far away for him to truly get an ID on them. Blue jays, though, a huge number of blue jays, like 25,000 blue jays, cowbirds, American robins, starlings, some rusty blackbirds, and then all other birds I kind of lumped together. So that's what we were finding with just this initial season of the morning flight counts. So it's pretty exciting. Now I'm going to talk about the next technique we use, which is bird banding. Many of you have heard about it or visited us or seen it in action, and this is a technique for studying migratory birds that's been going on for many decades and has been happening here at Presque since 1960. And basically we use these nets. They're called mist nets or specialized nets, and you do have to have a permit to use these. And you only get a permit if you have the right kind of training and experience. That's because they can be very deadly for birds. And elsewhere in the world, people actually harvest songbirds for food using these nets, and so that's not allowed in the US. But each time you catch a bird, you retrieve it out of the net, we bring it back to our station, we put a band on the leg. These are aluminum bands, they're each individually numbered, like a social security number with 9 digits. And there are records kept on all the bands that go out to the banders, and we have to, the banders all report back to the banding lab, what birds they put them on, where and when, and that kind of information. We do take some measurements on the birds, like the wing length, and we weigh the birds, look at their fat condition, identify the age and sex of the birds. So anyway, this is a process of was an early citizen science project actually, and nowadays it's less citizens and more technical. They don't, I guess the banding lab decided they didn't really like everybody just banding these birds in their backyards. So anyway, I was going to say the history of the banding here, there's been quite a bit of history. Like I said, since 1960, there are 2 permits from the banding lab with a lot of records from here. The Lieberman family or Lieberman family and the Stulls. Some of you may have known the Stulls. And so they had banded quite a few. The Liebermans banded something like 46,000 and the Stulls banded 25,000+. But they all stopped banding. Well, the Stulls stopped in '99. Ron stopped in 2006, and that's when I started. Basically, I came here and I have a banding permit, and Harry Leslie actually said, hey, it would be great if you could do the banding program, and I said, oh, absolutely, I love banding. So anyway, ever since then I've been doing it. On my permit, it's, we just had a whole other season, so I realized my number was outdated, and I just put a approximate number. That 131 species we've banded. We do have it open to visitors. We did not have it open in the spring of 2020. Luri Koich, many of you have met, is our primary bander, since I have to do other things too. I was able to track down a record. I was looking for the one from 50 years ago. This is what all the banders used to send in their banding reports to this journal called North American Bird Bander. And I found the record from the fall of 1981. Gene Stull sent this in just for reference. And like I said, this is long-term monitoring data. Here's what Presque, Pennsylvania, what they captured that fall between the 2 permits. And yeah, here's their list of top 12 species. Interestingly, white-crowned sparrows at the top. We don't get nearly that many of them anymore. So there's some interesting changes that have happened in terms of species diversity and abundances. But here's a comparison between the fall of 1981 and the fall of 2020, which we just ended on Sunday. They had actually only operated 14 days. They were mostly on weekends. They all were working at that point. We actually were able to operate 41 days. They used 20 nets. We used 15. But here's our unit of effort by which we want to look at our capture rates. To be able to compare then and now, we need to have it total birds divided by how much effort it took to catch those birds. We had a lot more missed net hours, that's the standard unit that's compared, than they did. Our largest daily catch for the fall was that, theirs was that. They only banded 660 birds, but we banded 2,298. Again, our effort was much higher than theirs. They had 58 species, and we had 68. But here's this birds per 100 net hours, which has been a standard unit of capture rate for mist netting. Theirs is 98 birds per 100 net hours. Ours is 68 birds per 100 net hours. And I just want to point out that, remember that 28% decrease? This is basic. I don't want to infer too much from a single data point here, but this is very much in keeping with that, those numbers. So I think that's all I'm going to say about the banding. We'll continue to do it. We welcome students to come help and volunteer. We'll be back out there in the spring. Now I'm going to talk about the very last part, and I know I'm the only thing between you and lunch, so. Yeah, I'm going to talk about now the tracking of movements of individual birds with radio tags. And This technique is different from the others in that it's not just about total numbers of birds, it's more about individual movements. Where are the birds actually going? This individual bird, we put a tag on it, we can figure out where the heck it goes. And so the questions you can ask are more like this. How important are the habitat patches on and off Presque Isle? How are the birds moving around through this urbanized environment? Are they using any urban areas or not? So that's, I'm going to briefly recap a little bit of previous work from 2017, where we really were looking at how these birds navigate this urban area, semi-urban area. So we actually had this type of little radio tag here that is approved for these birds. We looked at 2 species, black-polled warblers and Swainson's thrushes. And we took them and released them at several different locations off the park and then tracked them using hand-held antenna and estimated where they were based on that. But we're looking at whether they use different habitats differently and what happened in terms of, did they go towards the lake? Did they avoid the, what happened to them when we moved them to these locations? And just very briefly, the yellow circles are the release locations, more or less. All those little markers are places where blackpolls were detected. So we had, I've already forgotten the number of total birds there. I think it was 6 blackpolls, and they're, unfortunately, the colors don't show up very well. But basically, the blackpolls are totally comfortable, apparently, moving around. They're not staying in the parks where we released them at all. But in contrast, the Swainson thrushes they just stay put in the parks where we released them. So the ones that we got released in McClellan Park, this little square one here, they just stayed there. They didn't try to go anywhere outside of that until they totally left. The ones that were, the other place we released a Swainson thrush, that was a year we didn't catch very many Swainson thrushes in the spring, so we had relatively small sample size. This was up at Glenoto Camp. That bird just stayed there too, even though that was a more connected area, it could have moved around a bit. So I'm not sure what to conclude from whether it's the habit of the birds, they were able to find enough food right where they were, and they didn't need to move around, or whether they really were, did this bird really, was it constrained in some way by being in that park? But based on the fact that this bird did the same behavior, I'm thinking maybe that's really not the right conclusion. So that's what happened then. Very quickly into spring of 2021, before lunch here. To look in more detail about birds moving on and off of the peninsula, I was able to get funding to put up these new antennas on the side of these posts here. It's a little hard to see against the sky, but we put one fixed station here, right by the air monitoring equipment, but DEP owns, by the houseboat electrical outlets. So our antenna's up here now. And then the other one is across the canal, or the shipping lane, whatever, shipping, we call that big channel, that's the word. Other side of the channel, the South Pier. And so I have one down there too, thanks to the Port Authority, let me put it up on one of their posts. And so the idea was that if we release the tagged birds here at Fry's Landing, we could tell if they go across the channel onto the mainland or not. And so here's what we did this spring, a quick summary. We're using a different brand of transmitters. They're not the white ones anymore. We're using these black ones. We have to put, this is called a leg loop harness. You glue it on with super glue. Ultimately,